So a lot of engineering necessary, which need a lot of genetic innovation, where you can reasonably make a claim that you will need dozens or hundreds of coordinated mutations, and even for a single one you would need 10 times the time that is available based on the fossil record. And when this argument was presented in 2009 debate at Beverly Hills, Donald Prothero, who authored this famous anti-ID, anti-creationist book, Evolution, What the Fossils Say and Why It Matters, he was absolutely clueless how to respond to Rick Sternberg's argument, and he didn't even understand it. You can listen for yourself. I posted the link to the YouTube audio recording of this debate, and you will be astonished. Donald Prothero, the famous vertebrate paleontologist specialist for perissodactyls, was completely demolished by the intelligent design proponent and had no answer to this argument. He did not even understand it, which is, in a way, embarrassing.
The sequence of forms that I have above me, very close to very similar to a figure that you have in your book, Doctor. From the first impression of it is a nice transitional series, a nice progression of forms. You start out with a wolf-like or pachycetid ancestor, and then you end up with fully aquatic whales. And moreover, it's something that appeared in a well-known time frame. So you have this transition starting out from a true walking whale and ending up with a fully aquatic creature in a period of what? Roughly 9 million years. I'm taking one estimation, a monograph on Dorodon that said starting from Pachycetus, ending in Dorodon is a time frame of 9 million years. Well, the sequence that we see here, starting from a common ancestor and ending up with a derived form, is a pattern. It's a pattern to be explained. The morphing that we can imagine in our minds, in and of itself, is not a causal explanation. And you are right. What we have to do is provide testable hypotheses. And there is a testable hypothesis that has been proposed. It is that you have random mutation rates and recombination, and then you have selection coefficients, and what you get is incremental change over time, which for many people is the definition of evolution. Now, what's interesting is that in the past 2 years, several papers have been published, quite interesting papers that show that this nice progression of forms, and I don't question that progression of forms, by the way. I'm not a young Earth creationist, and I'm not an old Earth creationist, as insinuated, but that this progression of forms was rather more abrupt. Some of the sequence, for example, if you look in the middle, you've got a taxon like Gaviacetus and Protocetus and Georgiacetus. These are called protocetids, and they're depicted as having rudiments of tail flukes and reduced hind limbs, and essentially they look in between a walking whale, a terrestrial whale, if you will, and a fully aquatic whale. But what we now know is that from the skeletal evidence is that the transition took place from creatures that gave birth on land, that had well-developed pelvises, that had well-developed hind limbs, that had a sacral vertebra, to creatures that had highly reduced hind limbs, no sacral vertebrae, absent pelvises, and that gave birth in a fully aquatic environment. And moreover, if you take seriously, and I do, that this transition occurred from Pachycetus to a true marine whale in 9 million years, then this transition from these protocetids to fully aquatic whales was rather abrupt in a few million years. Now, what's interesting is that if you get a good textbook on cetacean anatomy and embryology, and you look at the development of these structures, or just some of the soft tissue parts that are necessary that you could never discern from looking at the bones, you know that to even have a basic fully aquatic whale in and of itself is a feat of engineering. I'm not saying intelligent design, I'm just saying engineering. What do you need? You need a countercurrent heat exchange because the testes are inside the body. You need a ball vertebra. Why? Because unlike a dog's tail that wags from side to side, you have to have a tail that moves up and down. You have to have the tail flukes. They're not found in the protocetids. They're only found in the fully developed aquatic whales. You got to have the musculature for it. You have to have the reorganization of kidney tissues. Why? because you're not drinking fresh water. You have to intake salt water. You have to nurse young underwater. You have to modify the mammary glands for that. The orientation of the fetus of, for example, the protozoes, the so-called intermediates, we know were like those of terrestrial mammals. However, in the case of whales, the orientation is reversed. You get reduction of the hind limbs, forelimbs transformed into flippers, hydrodynamic properties of the skin, special lung surfactants. When whales dive and they come back to the surface, the lung has to re-expand and it has to do so very quickly. Novel muscle systems for the blowhole, modification of the eyes, modification of the teeth, and it's on and on. So what do you have? You have, in a short window of time, hundreds, if not thousands, of character state changes. Dramatic reorganization. Now, my question is very specific. Can this geological pattern that I accept and that I'm not going to attribute to outside intervention of any form, can this geological pattern, this progression of forms, be explained by the processes of population genetics? And there, in other words, does neo-Darwinism, and just neo-Darwinism, provide an adequate causal explanation for the origin of these structures? Well, what is neo-Darwinian evolutionary theory? It's essentially that mutations generate variation. There are different kinds of mutations, different frequencies. Natural selection preserves the successful variants. And therefore, if new traits are necessary to produce a different biological form, depending upon the number of traits, then it's assumed that you have many adaptive mutations that had to arise and had to be preserved. All right? And how do you test this? Well, you can apply the equations of population genetics, and you can model them. You can get a textbook, and it's done all the time. I brought some of the literature with me. And you can determine the plausibility, whether or not there were enough mutational resources available for a given scenario to work. In other words, you can ask, was there enough mutational grist for the mill of natural selection? Now, it's a numbers game. What do you need? You need three necessary factors. Many generations, that is to get the number of traits appearing, and you have to have many generations and or rapid mutation rates and or large population sizes. One more requirement, that it's not enough just to get a mutation. It's not enough for a gene variant to appear in a population. It's got to be fixed. It's got to go from that initial infinitesimal copy, the lone individual out there, to becoming the norm that's established. If you do not have these, then you have two possibilities. One is that variation becomes exhausted in a lineage. And you see this, for example, with endangered species or populations that suffer from what's called inbreeding depression. Or you can have that, but you also have what's called genetic drift, which means that you do not have the population sizes necessary for natural selection to be effective. And the population literally drifts. It's a random or nearly random process. So, I assert, and I'm only talking about neo-Darwinism, and I'm not talking about anything else. I assert that there are three problems, and they're readily identifiable. One is that there were too few generations involved in the transition. I'll explain why. One is that the adaptive mutation rates, known adaptive mutation rates, are far too low. And the third is that mammalian population sizes are far too small. Now let's look first at adaptive mutations. I drew how I calculated this on a board, so should there be any questions, I'll be happy to walk you through it. What I calculated here, given a known rate of an adaptive mutation, this is a mutation that appears and doesn't just affect a trait slightly in a positive way, but it affects a number of traits and in a positive way. And we know these from organisms that we can study well. Bacteria, viruses, and fungi, like yeast. And what I did is I took three breeding population sizes. Think in your mind, these are walking whales I'm talking about. And all that matters are the individuals that reproduce. The rest you don't factor in. And I plugged in how long it would take for one adaptive mutation to appear in a population. What population size would you need? And what we can do, very simply, is we can rule out two of them. And here's why. If you assume that, say, these walking whale populations were the same size that we know of some very large animals, large mammals, for example, say 10,000 breeding individuals per generation, 5 years per generation, then we know, well, you can readily calculate, it would take around 136 million years for that to appear. But guess what? You have 9. And in the case for many of the trait changes, you have far less than that. If you assume a breeding population of 1 million individuals, you run into a problem, and here's why. With mammals, breeding population sizes are in the tens of thousands to the hundreds of thousands. House mouse. House mice exist in millions, if not billions, of individuals globally. And they're breeding and they're doing their business underground. But here's the thing. The breeding population size of the house mouse is around 600,000 individuals. It's between 410,000 and roughly 800,000. In our case, hominids, until very recently, the breeding population size was around 20,000 individuals per generation. So numbers matter. But so let's put that aside. But what if you needed multiple coordinated changes to affect the transitions that I've illustrated? Here going from protocetid, Georgiocetys, to a Dorodon, actually Basilosaurus. Well, let me give you an example of one. This is a cetacean countercurrent heat exchange system. Cetacea have their testes on the inside of the body, right next to the muscles that generate heat during swimming. Now, what happens in mammals is that if the testes are in the inside of the body and they're not cooled, you have sterility. So you have to cool down the glands. And this is done, similar to a modern refrigeration system, by shunning cool blood from the tail flukes to a plexus, or nexus, if you will, of arteries and veins that dissipates the heat from the gland. So even though a cetacean is moving in the water column, the testes are remaining below core body temperature. Now, let's assume, and I think this is an absurd assumption, that only 2 mutations were necessary for this complex adaptation that I've just shown. We can assume that it was far simpler than that. It doesn't matter. Yeah, coordinated mutations. Durrett and Schmidt, 2008, have an interesting paper in genetics, and they were attempting to refute Michael Behe's book, The Edge of Evolution. And they calculated the probability of getting 2 coordinated mutations, very simple mutations, not in a protein-coding gene, but just in some DNA sequence that, say, would bind a factor. Well, the probability and in various population sizes. And their conclusion was that, well, you could get these 2 coordinated mutations in the human lineage. You've only got 20,000 individuals on average per generation, once every 216 million years. Now, if you factor in that the human-chimp divergence was 5.5 million years, it seems that it's rather unlikely. For walking whales, if you adjust the equation to say, well, you're dealing with more individuals, that adds up to around 43.3 million years. That's just the paper and the calculations that they used to come up with that conclusion. So here's the nub of the issue, and the only issue I'm here to discuss tonight. If the evolutionary process, and by that I've defined it as neo-Darwinian theory, would typically produce only 2 coordinated mutations in 216 million years in a hominid lineage, and if in these aquatic mammal precursors, these whales, the evolutionary process would have only produced 2 such mutations in 43 million years, how could this process alone have produced fully aquatic cetaceans with their multiple anatomical novelties, requiring many hundreds, even thousands, of adaptive changes in less than 2 million years, even less than 9 million years, perhaps even 50 million years? Dr. Meyer, I will think, but if you have any questions about that? I'm saying it doesn't add up, yes. Thank you.
I've got everything in 5 minutes. Okay. I'm going to make an additional point. The idea of population genetics was hatched in the 1920s and 30s before we knew about the information revolution in biology. And as a result of that, it made a number of assumptions that are still with us today, and yet are not well founded. The main assumption it made was that it's easy to produce new traits by making with mutations. Genes were thought to be unitary entities prior to 1953. You change a gene, you automatically get a new trait. People didn't know how frequently they occurred, but it was assumed that it was easy to generate new traits by mutating genes. Since 1953, with the recognition that genes are actually sections of digital code along the DNA molecule, that assumption of population genetics has had to be revisited. And what it means is that as difficult as the problem is from the standpoint of neo-Darwinism and its mathematical expression in population genetics, the problem of generating new biological form, anatomical novelties, like all these cool structures that Rick was talking about in whales, is even more difficult. And in particular, what we've recognized is that building new complex animals does indeed require lots of new information. Not complexity, as Dr. Prothero referred to it, but information, specified arrangements of sequences. If you think about mutating something, and you realize that you need information to build a new organism, just like you need new information to give your computer a new function. And you begin to think about it in light of the information revolution, it raises some other questions about the power of mutation. Ask yourself a question. If you've got a section of digital code, a section of alphabetic text, and you begin to randomly change it, blindly, are you more likely to degrade the meaning and function that's there, or are you more likely to enhance it? Clearly, you're overwhelmingly more likely to degrade it. And one of the reasons for that is it turns out that the ratio of the number of sequences of alphabetic characters that perform functions or are meaningful to the sequences that don't is very small. In a 10-letter sequence in the English language, there is one functional sequence for every 10 to the 14th, or 100 trillion non-functional sequences. So inevitably, you start to degrade things, you're going to move the sequence away from function into non-function. Now, this was intuitively, biologists thought the same thing in the 1960s about mathematicians, in particular mathematicians and physicists at MIT, who began to think about the plausibility, the mathematical plausibility of mutational theory. But they didn't know how, if the same thing was true of genes and proteins, that the ratio of the functional sequences were very small. The ratio of function to non-function was very small. And this question was taken up building on the work of an MIT scientist named Robert Sauer by Douglas Axe, who is the director of the laboratory at the Biologic Institute, which is where much of the laboratory research that Dr. Prothero says is not done is done. Axe began his research at Cambridge University, and he asked a critical question. The question was this: how rare or common are the functional sequences of genes or proteins in relation to all the possible ways there are of arranging the base characters in the genetic message or the amino acids in the proteins? If it turned out that the sequences were very common, that functional sequences were common, it was possible to imagine that by mutation you could kind of skip from one island of function to the next very readily. But if it turned out that the ratio of functional to non-functional sequences was astronomically small, then what you would have is a situation where mutation would be searching indefinitely, trying to find the functional sequences and not having enough time to do it, like looking for a tiny, tiny pin in a gigantic haystack. Well, Axe, through this period, about 14-year research published in the Journal of Molecular Biology, determined that the ratio of functional proteins to the corresponding space of all the possible ways there are of arranging amino acids of that given length is astronomically small. The ratio is 1 in 10 to the 77th power. So to get a handle on that, imagine this. Imagine that you're trying to find the combination on a combination lock. But instead of having a combination lock that's just got 4 dials, or 10 as my artist has rendered, imagine you have one that has 77 dials with 10 settings on each one, and you have only one functional combination. Is it going to take you a little bit of time or a lot of time to search that space of all those possibilities to generate the new combination or information you need to unlock the lock? Obviously, it's going to take you an immense amount of time. And that is the measure to the measure of the probability of generating one protein fold, a domain of a protein, not even the whole protein, okay, in axis research. Now, to build a new Cambrian animal, to build a lens-focusing eye, a compound lens-focusing eye in a trilobite, you're going to need many genes. To build the whole animal, you're going to need many genes. Arthropods have about 50 cell types, each of which have many dedicated proteins, many of which would not have been present in the immediate ancestor to the arthropods in the arthropod prior to that critical explosion that I talked about in my opening remarks. Now, one more problem as far as the generation of information. It turns out that you don't only need information to build genes and proteins. It turns out to build body plans, you need higher levels of information, higher order assembly instructions. DNA codes for the building of proteins, but proteins must be arranged into distinctive circuitry to form distinctive cell types. Cell types have to be arranged into tissues. Tissues have to be arranged into organs. Organs and tissues must be specifically arranged to generate whole new body plans, distinctive arrangements of those body parts. We now know that DNA alone is not responsible for those higher orders of organization. DNA codes for proteins. But by itself, it does not ensure that proteins, cell types, tissues, organs will all be arranged into body plans. And what that means is that that body plan morphogenesis, as it's called, depends upon information that is not encoded in DNA, which means you can mutate DNA indefinitely. 80 million years, 100 million years, till the cows come home, and it doesn't matter. Because in the best case, you're just going to find a new protein someplace out there in that vast combinatorial sequence space. You're not, by mutating DNA alone, going to generate the higher order structures that are necessary to building a body plan. So what we can conclude from that is that the neo-Darwinian mechanism is grossly inadequate to explain the origin of the information we need to build new genes and proteins, and it's also grossly inadequate to explain the origin of novel biological form, which is to say, it's not adequate. Thank you.
Concluding statement, 5 minutes.
I think both the gentlemen opposite are insufficiently sensitive to the importance of mind in the cosmos. Our minds. We do lots of cool stuff. We know what a mind can do. We know what it means to be intelligent. As long as we are now debating intelligent design, let me respond to the points that were made. We didn't think this was a debate about we thought it was about the origins of life and evolutionary theory sufficiency, which meant it was mainly about the mechanism, the cause offered by evolutionary theory, which is natural selection and random mutation. But if we want to talk about the origin of life, I'm really happy to do that. I did just write a long book on the topic. Dr. Prothero mentioned in his opening statement, he talked about Stanley Miller and his synthesis of amino acids. Miller and many other prebiotic chemists repudiated the relevance of those experiments late in life because they realized that the conditions under which the amino acids were synthesized did not simulate the conditions on the early Earth. That was problem one. Problem two, when the amino acids were allowed to collect and sit around for a while, a sludge, a chemical sludge accumulated called melanoidin. In order to keep that reaction from taking place between desirable amino acid and byproducts, problem known as interfering cross reactions, intelligent investigators have to intervene and take out reaction products that would cause biochemical reactions to go in an unfavorable direction. That's the hidden hand of intelligence, in this case the scientific investigator, playing a role in moving life or chemicals in a life-friendlier direction. But notice what's happening. The scientific investigator thinks that this is all natural, but what's really going on is a mind is directing the process. Dr. Prothero also talked about these these experiments with RNA, where the investigator is literally sequencing the RNA gene. That's, again, intelligence creating information. What goes on here is that with the materialistic mindset that has grown up around science, that scientists they're looking at nature, but they're not noticing their own intervention in what intelligent minds do. And when you step back and notice that minds have certain powers that material entities do not, you can begin to make inferences about the activity of mind in the past. Now, Dr. Schirmer raised the whole question of who designed the designer. One of the chapters in my book that I'm happiest about is the chapter 17 in which I take that on. I'll give you a brief synopsis of my response to that. I agree with him that one of the problems with invoking an immanent intelligence, an alien within the cosmos, is that it does lead to this kind of infinite regress. That's one of the reasons I favor the theistic design hypothesis over the spaceman hypothesis favored by, for example, Richard Dawkins, or at least tendered by him at the end of the movie Expelled, or tendered by Francis Crick. Another reason I favor the theistic design hypothesis is that there's also evidence of design from the very beginning of the universe and the fine-tuning of the laws of physics and chemistry, something I agree with Francis Collins about. When people say, well, who designed the designer, they're just, again, privileging materialistic thinking, materialistic explanations. You can say the same thing of the evolutionary hypothesis. So you accept the neo-Darwinian hypothesis, you can ask who evolved the evolver. If you concede, as many evolutionary biologists have done, such as Richard Dawkins, that there is no adequate undirected evolutionary explanation for the origin of the first life, you do the same who designed or who evolved the evolver infinite regress question on him. He accepts biological evolution, but it can't get started without chemical evolution, and he accepts that there isn't such a process. The real point is that in any metaphysical system, and now I'm not talking about scientific reasoning, but in any ultimate metaphysical explanation has to go back to some primitive, something from which everything else comes. And the question in modern philosophy of science is what is the best candidate to be that primitive, to be that thing from which everything else comes. I think post Big Bang theory, post the solutions to the field equations of general relativity, implying that you have a true singularity at the very beginning of the universe, in which the spatial volume of the universe curves on itself so tightly that you have a true singularity. I think the best candidate is to be that thing from which everything else comes is not a material entity. How much space can you put in zero spatial volume? None. I mean, how much matter can you put in zero spatial volume? So I think with the new cosmology, you can ask the question, which of these two metaphysical systems provides a better starting point? I think that's one of the reasons I'm a theist, but we weren't debating that tonight. We were just debating the adequacy of the neo-Darwinian mechanism, that is to say, modern evolutionary theory.